Diarrhea. For some of us, it comes as the result of enjoying a really greasy burrito. But for others, it's actually a matter of life or death. My specific project focuses on one rogue strain of E. coli called EPEC. Now, EPEC causes diarrheal diseases. And the most interesting thing about EPEC, what makes it different from normal E. coli, is the fact that it uses the most unsuspecting weapon to cause damage. It doesn't use like a sword or nunchucks, but instead it uses a syringe. So this syringe is actually a part of EPEC's body. So once it makes its way down to our gut, it'll find our intestinal cells. Here it'll stab them, and now that it's poked a hole in them, it's going to introduce a cocktail of proteins into these cells. Now, this cocktail of proteins is not a very good thing, because it turns out that these proteins are master hijackers. So once inside of our cells, they're reprogramming that environment, and what they're doing is causing our cells to now be more favorable to allow EPEC to grow, spread, and thrive. And this, of course, is the opposite of what we want, especially considering the fact that there are no treatments and no vaccines against EPEC. And current medical advice for infection with EPEC is just to rehydrate the patient and let the infection pass. But what if that infection doesn't pass? Especially for mothers in developing nations. Instead of getting to spend that first year of their child's life teaching them to walk and talk, they're now spending that first year in a hospital room with their child hooked up to an IV. But this doesn't have to be reality. What if we hijacked the hijackers? Think of it like this. If we can learn to understand how these hijackers actually work and why they're released from the syringe at all, then surely there can be a way that we can stop them from ever being released, right? And at a large scale, this means preventing the spread of EPEC infection. So my specific project focuses on understanding how I can stop these protein hijackers from even docking onto the needle so that they're never released. Our lab currently has data that tells us that there's a mystery protein that tells EPEC when it should send out this army of hijackers. And once we find out what that protein is, we'll be able to find it, turn it off, prevent this hijack, and finally have a fighting chance against EPEC infection. Thank you.